Hello, welcome to Biology, the scientific study of life. Blood vessels associated with the nephrons. Each nephron is supplied with the blood by an afferent arterioles, a branch of the renal artery that subdivides into the capillaries of the glomerulus, as shown in this figure. Notice that the capillaries converge as they leave the glomerulus, forming an efferent arteriole. This vessel subdivides again, forming the pretubular capillaries, which surround the proximal and distal tubules. More capillaries extend downward and form the vis recta. The capillaries that serve as loba of Henle. The vis recta also from a loba with descending and ascending vessels conveying blood in opposite directions. Notice that. Although the excretory tubules and their surrounding capillaries are closely associated, they do not exchange materials directly. The tubules and capillaries are immersed in interstitial fluid, through which various substances diffuse between the plasma within capillaries and the filtrate within the nephron tubule. This exchange is facilitated by the relative direction of blood flow and filtrate flow in the nephrons. From blood filtrate to urine, a closer look. Notice that in this section we will concentrate on how the filtrate becomes urine as it follows through the mammalian nephron and collecting duct. The circled numbers correspond to the numbers in this figure. 1. Proximal, one stage, a proximal tubule secretion and reabsorption in the proximal tubule substantially alter the volume and composition of filtrate. For example, the cells of the transport epithelium help maintain a relatively constant pH in body fluids by the controlled secretion of H positive. The cells also synthesize and secrete ammonia, which neutralizes the acid and keeps the filtrate from becoming too acidic. The more acidic the filtrate, the more ammonia the cells produce and secrete, and the urine of a mammal usually contains some ammonia from this source, even though most nitrogenous waste is excreted as urea. The proximal tubules also reabsorb about 90% of the important buffer bicarbonate, HCO3-, drugs and other poisons that have been processed in the liver, pass from the pretubular capillaries into the interstitial fluid and then are secreted across the epithelium of the proximal tubule into the nephrons lumen. Conversely, available nutrients, including glucose, amino acids, and potassium K positive, are actively or passively transported from the filtrate to the interstitial fluid and then are moved into the pretubular capillaries. Notice that. One of the most important functions of the proximal tubule is reabsorption of most of the NaCl salt and water from the huge initial filtrate volume. Salt in the filtrate diffuses into the cells of the transport epithelium, and the membranes of the cells actively transport Na positive into the interstitial fluid. This transfer of positive charge is balanced by the passive transport of Cl- out of the tubule, as salt by the passive transport of Cl positive or minus out of the tubule. As salt moves from the filtrate to the interstitial fluid, water follows by osmosis the exterior side of the epithelium has a much smaller surface area than the side facing the lumen, minimizing leakage of salt and water back into the tubule. Instead, the salt and water now diffuse from the interstitial fluid into the pretubular capillaries. 2. Descending stage 2. This one, descending limb of الوحدة. Notice that. Reabsorption of water continuous as the filtrate moves into the descending limb of the الوحدة. Here the transport epithelium is freely permeable to water but not very permeable to salt and other small solutes. For water solutes, for water to move out of the tubule by osmosis, the interstitial fluid bathing the tubule must be hyperosmotic to the filtrate. The osmolarity of the interstitial fluid does in fact become progressively greater from the outer cortex to the inner medulla for the kidney, thus filtrate moving downward from the cortex to the medulla within the descending limb of the الوحدة continuous to lose water to interstitial fluid of greater and greater osmolarity, which increases the solute concentration of the filtrate. 2 or 3 ascending limb. Notice that 3 thick segment of ascending limb. Ascending limb of the الوحدة, the filtrate reaches the tip of the loop, deep in the renal medulla. In the case of juxtamedullary nephrons, then moves back to the cortex within the ascending limb. In contrast to the descending limb, the transport epithelium of the ascending limb is permeable to salt but not to water. The ascending limb has 2 specialized regions, a thin segment near the loop tip and a thick segment adjacent to the distal tubule. As filtrate ascends in the thin segment, NaCl, which became concentrated in the descending limb, diffuses out of the permeable tubule into the interstitial fluid. This movement increases the osmolarity of the interstitial fluid in the medulla. The exodus of salt from the filtrate continuous in the thick segment of the ascending limb, but here the epithelium actively transports NaCl into the interstitial fluid by losing salt without giving up water. The filtrate is progressively dilated as it moves up to the cortex in the ascending limb of the loop. 4. Distal tubule. Okay, notice the stage 4, distal tubule. Notice that the distal tubule plays a key role in regulating the key positive and NaCl concentration of body fluids by varying the amount of the key positive that is secreted into the filtrate and the amount of NaCl reabsorbed from the filtrate. Like the proximal tubule, the distal tubule also contributes to pH regulation by the controlled secretion of H positive and reabsorption of bicarbonate HCO3-. 5 stage or 5 notice the collecting duct. This is the stage. The collecting duct carries the filtrate through medulla to the renal pelvis by actively reabsorbing NaCl. The transport epithelium of the collecting duct plays a large role in determining how much salt is actually excreted in the urine, though its degree of permeability is under hormonal control. Under hormonal control, the epithelium is permeable to water. However, it is not permeable to salt or in the renal cortex to urea. Thus, as the collecting duct traverses the gradients of osmolarity in the kidney, the filtrate becomes increasingly concentrated as it loses more and more water by osmosis to the hyperosmotic interstitial fluid. In the inner medulla, the duct becomes permeable to urea because of the high urea concentration in the filtrate. At this point, some urea diffuses out of the duct and into the interstitial fluid away along with NaCl. This urea contributes to the high osmolarity of the interstitial fluid in the medulla. This high osmolarity enables the mammalian kidney to conserve water by excreting urine that is hyperosmotic to the general body fluids. Back to the nephron and collecting duct regional functions of the transport epithelium. Proximal tube, tubular and descending limb of loba of Henle, and notice Henle, and distal tubular and thick segment of ascending limb and thin segment of ascending limb, and 5-1 stage is collecting duct. Notice also the circulation of filtrate, H2O, salts NaCl and others, HCO3. Notice also H positive, urea, glucose, amino acids, some drugs. Notice also the key. Red one is active transport, and the purple one is passive transport. Notice that. Okay, thanks for listening.